Hello everyone, thanks for watching this video. My name is Arash Bayat and I'm from Health and Biosecurity at CSIRO. Today I'm going to talk about understanding complex phenotype with BitEpi and EpiExplorer. A complex phenotype might be driven by many biological pathways. Some of those are controlled by individual genes, some others are controlled by multiple genes in a complex way. When multiple genes involved in the same biological pathway, we say there is an epistatic interaction between them. We expect to see some statistical relevance between the phenotype and those individual or interactive genes. Finding these interactions are computationally complex. Let's say you only have 100 genomic variants. There are 5,000 two-way combinations and 160,000 three-way combinations. The number of four-way combinations almost reached 4 million. Now, it's just for 100 genomic variants. Imagine a whole human genome data, which include tens of millions of genomic variants. It's impossible to test every single combination. BitEpi is the software that performs association tests on every single variant, as well as two-way, three-way, and four-way combination of variants. It performs exhaustive search, meaning that it tests all possible combinations, and it uses fast bitwise algorithm and accurate association tests, which we will talk about them later in this video. We just said it's impossible to perform exhaustive search on a large-scale dataset. Definitely, there should be some filtering that we select a number of variants before analysis. There are non-exhaustive search as well, but they are yet far slower than what can perform analysis on the whole dataset. We still need filtering variants. Note that when we filter variants or perform non-exhaustive search, we actually apply heuristics or approximation to our analysis, meaning that there might be some important interaction that we will miss. Regarding the first step of approximation, how much we have to reduce the dataset depends on how fast we can process data. For the exhaustive search pipeline, being fast is critical because it means you can take or process more data within the same computational budget and ultimately discover more interactions. Non-exhaustive search are usually faster and they can process more data in the same time. But note that they internally apply another layer of approximation which reduces the chance of discovering interactions. Before we skip this slide, I would like to introduce you to VariantSpark as well. That's another great software that we build in our team. It uses machine learning to rank variants in such a way that if you pick the top ranked variants, most of the significant interaction in your dataset appears between those variants. Complex interactions are the most difficult to be captured. When the interaction is complex, individual genes in the interaction may not show any statistical relevance to the phenotype. Even subset of interactive genes may not seem relevant. The interaction is only visible when you look at all the interactive genes together. That makes them very difficult to be identified. All exhaustive search programs test all possible combinations of variants within the dataset. So they are all pretty the same from this perspective. What it really matters is what they do at every single test. There are two steps in every test. Compute contingency table, which is basically the number of samples in different cohorts with different genotype pattern for the selected variants in the combination. And then compute statistics from that contingency table. BitEpi uses a novel bitwise optimization to speed up the first step, and in the second step it uses new entropy-based metric, which is more accurate detecting interactions. We compare BitEpi with a number of other exhaustive search programs. Boost for the first time uses bitwise optimization to speed up two-way pairwise interaction. It uses log linear model in the second step as a screening test. MPI3SNP uses the same bitwise approach but expanded to three-way search. And it also replaced the log linear model with an entropy-based model for the second step. BitEpi introduces a new bitwise optimization, which is perfect for 3- and 4-way interactions, or basically higher order interactions, which has been less discussed because they're high computational cost. We also include MDR because this is the only other implementation that performs 4-way exhaustive search, but it does not implement any sort of bitwise optimization. BitEpi is 1.7 times and 56 times faster for 3- and 4-way search compared to MPI-3SNP and MDR. The huge speed up compared to MDR is because MDR does not implement any bitwise optimization. Also, the statistic that we use in BitEpi is 1.3 and 2.1 times more accurate when detecting 2-way and 3-way interaction, and that is in comparison with Boost and MPI-3SNP. We figure out this number using synthetic datasets. BitEpi computes two statistics for every combination of variants, beta and alpha. This might not be the best visualization of them, but perhaps the easiest one, where each circle shows a variance and the overlap between the circles shows the interaction. So beta focus on combined association power of all variants, while alpha focus on the interaction effect size. Alpha is really efficient filtering out false positives, especially cases where the combined association power is high, but the interaction effect size between those variants are not that much. Getting back to original problem of complex phenotype, where there are many biological pathways and so many genes involved, many interactions and many individual genes, once we compute all these statistics, how we can infer information out of that? EpiExplorer is the solution that we built for this problem. EpiExplorer takes all the statistics that BitEpi produced for individual variants and interactive variants, 2-way, 3-way, and 4-way, and also all the genomic annotation you have relevant to your dataset, and then it turns them into a sidescape graph where there is an endless opportunity for you to plot data in different shapes in a way that you can make discoveries out of it. Sidescape is a dynamic and interactive environment. Definitely you can play with your plot, you can move nodes around, you can click on the nodes or lines and get all the information relevant to those pieces of the plot. And it becomes really handy when you want to understand large amount of interaction which are discovered by BitEpi. So firstly, I should mention that with EpiExplorer, you have the option to choose different visual elements like shape, color, size, to represent different genomic or statistical feature of the data, for example, of the association power or which chromosome they belong to or which genomic function the variant has. Like you can color microRNA and the protein coding gene with different color, for example. And there are two main plotting modes. So in the first mode on the left hand, you have all your variants as node and then lines between the nodes represent interactions. It's easy for two-way interaction as I highlighted in yellow, which we connect two variants. But then for 3- and 4-way interactions, you will see we struggle to represent them with line because we can't simply do it with one single line. So we create the second plotting mode on the right where you can see interaction as node as well, but definitely with different shape or color so that you can distinguish them from your variant node. So the first mode is perfect when you want to study like which region of the genome interact with each other. We are not really concerned about like the order of interaction. And the second mode is great when you focus on the interaction themselves. One example plot that you can create is that you may lay out all the variant nodes on a circle sorted by their genomic position, and then From there, you can easily see which part of the genome interact with which other part. Like the small plot that we provide here is for synthetic data, but it shows how efficiently you can see different regions of the genome are connected to each other. Another feature, as I mentioned, is that you can change the thickness of the interaction line based on their statistical relevance to the phenotype to be able to see the most important one and focus on those. Just to mention that in a Cytoscape environment, you can hover over all these lines and nodes and see all the information relevant to them. So there's also a great filtering feature in the EpiExplorer, which doesn't come with the Cytoscape, where you can hide some of the nodes or bring them back while keeping all other nodes at the same place. And that's very important because when you have a large plot, you already study the plot, and then you want to change it a little bit. If the layout has been changed and nodes move, then you lose all the information you already gain about the plot. But in this example, for example, let's say we have variants on chromosome 6 and then all the variants in the protein coding gene. Now we remove everything except those in chromosome 6, and then we bring back all the protein coding genes, and at the end you can gray out all the variants which are not in protein coding genes, and you end up with this plot with one single interaction highlighted. So this was a made up example just to try to explain you what sort of things you can do with this EpiExplorer, and it was only for the first type of plot. There are many other ways you can plot and filter and process your data here in EpiExplorer, and it might help you to understand details which are very difficult to appreciate when you look at the whole data just in a messy environment. If you already have a GWAS dataset, there might be some interactions waiting for you to discover them. So try BitEpi and EpiExplorer on your dataset and see what you can find out with your data when you consider those higher order interactions. Thanks again for your attention. I hope you enjoyed this presentation.